Good morning, my fellow teammates and all the audiences that I appreciate who will watch this presentation. On this blissful occasion, we from the small group discussion, B3 group would like to present our student project entitled The Role of Calcium in a Nerve Transmission. But before moving forward to the next plan, let me introduce all members of this group, which is shown in the slide. Next. We will present our student project with three main chapters, which are introductions, literature review, and closing. For the first speakers, time is yours.
Hello everyone, my name is Gabriella Ferenina with 081 as the last three digits of my student number. This time I'm going to explain about background of the role of calcium in nerve transmission. The nervous system consists of the central nervous system and the peripheral nervous system. The central nervous system, brain and spinal cord, is the center of command. Meanwhile, the peripheral nervous system, or nerve, has the responsibility to connect every part of the human body with the central nervous system. When the human body is exposed to a threat, the body will immediately avoid that threat. That threat will cause sensory nerve to send an impulse to spinal cord. The spinal cord will send an impulse to certain muscle via motoric nerve to avoid the threat. The transmission of the impulse from one neuron to another neuron is called nerve transmission. Calcium is one of the most essential minerals for humans. The primary role of calcium is to move muscle. It is primarily found in bones and teeth, although it can also be injected to blood if necessary. In nervous system, central nervous system use calcium as a crucial signaling chemical and ion in many different activities, such as gene expression, synaptic transmission and plasticity, neural excitability, and cell maintenance. Not only is proper modulation of calcium signaling essential for neural physiology, but it also essential for cell survival. This all from me. The next will be continued by my colleague. Thank you.
Hi, my name is Putu Sri Dianingsih and my three last digit number of my ID is 070. I will continue the presentation. So the problem formulation of this student project start first, what is calcium? Second, what is nerve transmission? Third, how does neurotransmission work? Fourth, what role does calcium have in neurotransmitter release? And the last one is what factors can cause disease in neurotransmitter release. The next one is purpose. The purpose of the student project start first, to give a comprehension about calcium and nerve transmission. Second, to explain the mechanism of nerve transmission. Third, to give information about the role of calcium in neurotransmitter release. And the last one is to inform about factors that can cause disease in neurotransmitter release. The last one is benefits. The purpose of this literature is to present the proper information about the role of calcium in nerve transmission. Please. This literature is also intended to give the readers new insight about the mechanism of nerve transmission. We sincerely hope that this literature is going to give a good knowledge of the role of the calcium in nerve transmission and will contribute to the better improvement of the treatment for neurodegenerative disorders. For the next presentation, I will pass it to my partner.
Okay, I will continue this presentation by presenting the literature review, which consists of definition and biochemical properties of calcium, definition of nerve transmissions, mechanism of nerve transmissions, and the role of calcium in a neurotransmitter release. I am Wulan with three last digit numbers, 86, and I will deliver the definition and biochemical properties of calcium. Calcium is a protein mineral involved in a various biochemical process throughout the body. It is essential for appropriate heart function, bone structural integrity, and muscle contractions. It is also confers as an enzymatic signal in the biochemical pathway and plays a role in maintaining the nervous system. Calcium is tightly regulated by the parathyroid hormone, calcitonin, and calcitriol, which is metabolism is characterized by a sequence of homeostatic feedback loops designed to reduce overreactions and keep variable within normal limits. Rapid release of mineral from bone is essential for maintaining adequate serum's ionized calcium level. Next slide. The largest calcium is stored in the endoplasmatic reticulum, especially in the sarcoplasmatic reticulum in the sartorial muscles. On the physiological stimulation, calcium is detached from the RA and further stimulates calcium influx from the plasma membrane. Specific protein and the canals also redound to customize regulation of the entire cycles. Calcium channels plays a more involved role in the cells by enabling it to generate many cellular responses, as calcium has many roles in the body. Therefore, calcium channels comes in abundant and always include all calcium permeable pore-forming membranes protein. Thus, it can be used for the transport of these ions across cell membranes. For the next speaker, the floor is yours.
Hello everyone, my name is Nathanael Farrel with 077 as the last three digits of my student number. This time, I'm going to explain about nerve transmission. Neurons are cells that possess some unique abilities. These cells become stimulated as a result of their polarized membranes. Additionally, the membranes of the neuron has many kind of ion channels that selectively permit the passage of particular ions. Ion channels are required for the passage of ions into, in, or out of neurons. When a neuron is stimulated by pressure, heat, light, or chemical information from other cells, the neuron becomes activated. Nerve transmission happens as action potentials propagate through the neuron and synapse, causing a release of neurotransmitter in the synaptic cleft. Some synapse connects a neuron directly electrically, but chemical synapses make up the majority of synapses. It is more complicated for nerve impulses to travel through chemical synapses, that is, with the help of neurotransmitters. That is all from me. The next will be continued by my colleague. Thank you.
Hello everyone, my name is Natasha Putri with 083 as the last three digits of my student number. Now, I will explain about the mechanism of nerve transmission. Before a nerve transmission occurs, there must be an impulse for the transmission to conduct. When the nerve impulse reaches the axon end, some chemicals released from the neurotransmitter will help the transmission. This procedure, called synaptic transmission, can happen electrically or chemically. Neurons that are close enough together can electrically transfer ions from one neuron to another, whereas on chemical synapses, the presynaptic neuron releases neurotransmitters from the storage vesicles into the synaptic cleft, which will bind to postsynaptic neuron. Next. Now I will proceed with the steps of synaptic transmission. The first step is intracellular vesicles loaded with neurotransmitter are targeted to the presynaptic membrane. Second, an action potential triggers exocytosis to release the neurotransmitters to the synaptic cleft between two neurons. This exocytosis is caused by CA2 plus triggered membrane fusion. These occurs very rapidly or in less than 1 millisecond and is dependent upon an influx of calcium ions. The last step is neurotransmitters bind to the receptor on the presynaptic membrane to activate the signal transduction pathway. So that's all from me. Next will be proceed by my colleagues. Thank you.
Okay, thank you for the time that has been given to me. Introductions first. My name is Wilson Ekaputra Tanuwidjaja, with the last three digits of my student number being 084. And now I'm going to start explaining about the role of calcium in neurotransmitter release. So, calcium ions hold three main important roles in nerve transmission, which are in exocytosis, endocytosis, and in axonal transport. We shall start with exocytosis first. So, exocytosis is a process where vesicles fuse together with the cell membrane to release part of its vesicle load. So, exocytosis is caused by the influx of calcium ions that happens in a specialized presynaptic structure called the active zone, or we can simplify as AZ. So, before the calcium influx and exocytosis occur, the vesicle containing neurotransmitters must be in a ready-to-release state through three processes called trafficking, docking, and priming. And I will start with explaining what trafficking is. So in the trafficking process, a protein called synapsin-1 is found on the vesicle membrane and is this phosphorylated state and binds with the cytoskeleton elements, including actin. On the vesicle membrane, another protein called CAM or calmodulin-dependent protein kinase 2 is also present there. And during the trafficking process, after the vesicle is able to mobilize, motor proteins then carry the vesicle along the microtubule and actin filament tracks. So after the vesicle has arrived, and the following process called docking takes place. So here are our sources document below, and here's a picture depicting the process of CAM exocytosis. That is all from me. I'll let my next colleague continue.
Hello everyone, my name is Ariawan with 073 as the last three digits of my student number. This time I'm going to explain about the next step of exocytosis. The next step after trafficking is docking. The docking process involves the VAMP protein, plasma membrane proteins, a cytoplasmic protein, and AZ protein. When the vesicle is brought into close contact with the plasma membrane during the trafficking process, the plasma membrane protein called syntaxin, the VAMP protein called synaptobrevin, and the cytoplasmic protein called SNAP25 interact with each other. The formation of the snare complex indicates the end of the docking process, which will take the vesicle to the following process called priming. Next. Priming refers to a formation of pre-fusion complex in an ATP-dependent manner that will allow the vesicle to quickly respond to calcium influx. The key proteins of this process are MUNG13 and MUNG18. MUNG13, in cooperation with MUNG18, promotes and forms a parallel configuration of the snare complex. That is all from me. The next step will be continued by my colleague. Thank you.
Hello everyone, my name is Joanna with 079 as the last three digits of my student number. This time I'm going to explain about the fusion step of exocytosis and the role of calcium ions in endocytosis. Now, continuing from the priming step, the snare complex actually drives the fusion between the vesicle and the plasma membrane. This membrane fusion is triggered by the boost of calcium ions when the cell is excited. In order for calcium ions to trigger the fusion step, it requires a calcium ion sensor protein, which is the synaptotacmin protein. When the calcium ion binds to synaptotacmin, the snare complex will interact with the syntaxin protein to make the snare complex twist itself. This twisting motion helps fuse the lipid bilayers of the vesicle membrane and the plasma membrane. Next, please. Now, in maintaining the effectiveness of nerve transmission and the structure of the synapse, endocytosis must occur in order to clear the site of vesicle fusion. In terms of the role of calcium ions in endocytosis, it is known that calcium ions modulate the time course of endocytosis. Besides that, calcium ions play a rather significant role in synaptic vesicle, or SV, endocytosis. Now, calcium ion is involved particularly in bulk endocytosis. Bulk endocytosis is activated when there is a higher intensity synaptic activity that requires additional SV reuptake. This process relies on calcineurin, which is a calcium ion dependent protein phosphatase. Actin will be required in the invagination to form a large area. SV proteins transported by this method include VAMP2, synaptophysin, and vesicular glutamate transporter. That is all from me. The next will be continued by my colleague. Thank you.
Hello everyone, my name is Putu Sri Partha. The last three digits of my student number is 0. So I will explain about mitochondrial transport. The axon potential travels along the axon to the terminal axons, followed by exocytosis, which is all of this process requires energy. With an axon length that can even reach a meter in length, the method of sending energy by diffusion is certainly not enough. Therefore, axonal transport will transport mitochondria so that they are motile along the axon. Mitochondrial motility is regulated by concentration of a calcium ion. MIRO is a receptor that has two EFN CA2 binding site. MIRO is a receptor that functions in requiring kinesin to the mitochondrial membrane. At the excited presynaptic terminal, there will be an increase in intracellular CA2 concentration, which bond to the EFN of MIRO. This will cause key F5 transport engine to be disassembled and inactive. In this condition, the mitochondria become immobilized so that they can concentrate energy production for presynaptic activity. Mitochondria will be able to motile again when the intracellular CA2 plus concentration decreases. That's all from me. The next material will be explained by my colleagues. Thank you.
Okay, thank you for the opportunity. My name is Putu Chandrika Rahma Dewi with the last three digit name 71. So in here I will present about the conclusion of the student project. The nervous system is responsible for regulating and coordinating every activity that occurs in human body. The transmission of the impulse is called nerve transmission. In order of that, calcium plays a crucial role in signaling of chemicals and ions in many different activities such as for transmitting information. The transmission could send out an electrical impulse, and as the result, action potential propagates through the neuron synapse, causing the release of neurotransmitter in the synaptic cleft and then transmitted to the neighbor neuron to communicate with the rest of the body. Calcium ions also hold three main important roles in nerve transmission called exocytosis, endocytosis, and for mitochondrial motility. Exocytosis is a process where vesicle fuse together with the cell membrane to release part of its vesicle load and caused by the influx of calcium ions which happen in active zone. This process includes trafficking, docking, and priming. Endocytosis is a transport method with calcium ions in extracellular concentration modulated in the time course of endocytosis. And last, mitochondrial motility is carried out by the mitochondrial row GTPase or MIRO, which has two IF-hand calcium ions binding domain. So that's all for the conclusion. Thank you.
Hello everyone, my name is Sara Putri Bungaran with 078 as the last three digits of my student number, and this time I'm going to explain about the discussion of the student project. This discussion will explain several factors that can interfere with calcium's performance in nerve transmission that can lead to neurological diseases. The first one is changes in the regulation of protein phosphorylation, cytoskeleton dynamics, and gene expressions render neurons more susceptible to an increase in reactive oxygen species or ROS, which ultimately results in calcium-induced disruption of nerve transmission. And the next factors is cannabinoids or adenosine that has been shown to have an effect on LTP by facilitating strong LTP and inhibiting weak LTP. As a result, this interaction will affect brain connectivity to memory processes and interfere with memory consolidation. And the last factor is irreversible exocytotic injuries caused by massive calcium entry into neurons via N-methyl aspartate receptors, or known as NMDARs, as a result of an increase in extracellular glutamate, as well as other mechanisms by degradative enzymes. This increase in systolic calcium contributes to nerve cell death. And that's all from me. For the next speaker, time is yours.
Hello everyone, my name is Yuda with 096 as my last three digits of student number. At this time, I will continue the explanation about the discussion. Intracellular calcium signaling can have an impact on neurological disorder as well. There are broad examples of the disease that caused by intracellular calcium signaling disorder. The first one is Alzheimer's disease or AD. AD is a neurodegenerative disorder that impairs the memory formation and storage. This caused by the calcium I homeostasis dysregulation with high levels of calcium. The second one is Huntington disease or HD. HD is caused by an elongation of the N-terminal GAG trinucleotide stretch of the Huntington gene or HTT. The cause of HD is the dysregulation of calcium high homeostasis mediated primarily by NMDAR activation. The third one is Parkinson's disease or PD. PD is neurodegenerative disease characterized by motor symptoms. Mitochondrial dysfunction as a result of an imbalance of calcium I signaling is the cause of Parkinson's disease. And the last one is schizophrenia as a psychiatric disorder that caused by critical roles in calcium I homeostasis. That's all from me. For the next speakers, the time is yours.
Okay, so in here I will present about the advice for the student project. Based on the benefit of the calcium, the general public should be able to consume daily calcium intake properly to avoid some disease caused by calcium deficiency. And also, for the next researcher, to develop further research about the role of calcium in the human body, especially related to nerve and transmission, so that the general public can be more aware about the roles of calcium and the nerve transmissions. Okay, that's all for the advice. Thank you.
That's all from our presentation about our student project. We would like to apologize if there are any mistakes we made during the presentation. And thank you for your attention.